everyone. Good morning. I'm excited to welcome our speaker today, Dr. Kris Wood, who is currently an associate professor in the Cancer Center and Pharmacology and Cell Biology departments at Duke University. He did his Ph.D. in chemical engineering at MIT before moving all the way across the street to pursue a postdoc at the Whitehead and Broad Institutes. He and his lab have authored seminal papers in cancer therapy resistance, discovering novel genetic dependencies, and uncovering new effective combination therapies in cancer. He's a field leader in targeted cancer therapy and tumor evolution, and we're delighted to host you today. Please join me in welcoming Dr. Kris Wood.
Okay. Well, thank you very much for the invitation and the opportunity to come here. I have to say, this is the coolest seminar room I've ever seen. This is really, really, really cool. I'm going to try to speak at the right volume, but if you guys hear me talking too loud or too softly, let me know. I can't quite gauge. My lab studies tumor dependencies, which I think everyone here knows are the genes and pathways that cancer cells require for their survival. And of course, we're mainly interested in dependencies that are somehow or another selective to cancer cells relative to normal cells. But there are a lot of labs that study cancer dependencies. And I think the thing that probably distinguishes us from most of those other labs is the fact that we tend to think about dependencies as entities that evolve with the tumor. Right? So the dependencies that are present at the moment a tumor is diagnosed may disappear over time, right? And we all know that's true because we see drug resistance that emerges. But it's also true, and to us really interesting, that there are sets of dependencies that arise during tumor evolution in deterministic ways. And so in the talk today, what I'm going to be spending most of my time on is this concept, which I think is a lot more unexplored in the literature, but which could give us in the future the ability to build therapies that can potentially shape tumor evolution in more interesting ways than current therapies do. So just very briefly, these are my financial relationships and in the current and the past. One of the ways of thinking about our work is thinking about an index patient like the one who's shown here. So this patient, as you can tell, at the point of diagnosis, is diagnosed with metastatic disease. They have multiple tumors in their bodies. And if you look at some measure of clonal diversity, let's say in this case it's genetic diversity, you will see that there is heterogeneity, right? That there are clonal and subclonal populations within that tumor that presumably exhibit different phenotypic properties associated with their different genetic properties. Now, if this patient is matched with an effective therapy, what you would expect is two things. One, the total disease burden in their body would decrease. And two, there would be a shift in the clonal distribution of cells within the tumor, reflecting differential fitness in the therapy environment. And if the patient were to continue on therapy, I'm going to use this, were to continue on therapy, then what you might expect is that eventually their disease would progress, there would be an expansion of disease in their body, and there'd be further shifts in the clonal distribution of cell types or cell states in the tumor. So in our lab, we study and are interested in dependencies at each of these three phases of disease evolution. But in the talk today, I'm really going to focus on dependencies that emerge during tumor evolution at the point of maximal response and then at the point of resistance and progression. Okay, so first, what are the dependencies that emerge in residual disease, which is another word for the point of maximal response? So we don't know all that much about the biology of residual cancer cells, the cancer cells that survive therapy. We sometimes see schematics like this one, which refer to these surviving residual or drug-tolerant persister cells that survive therapy and that serve as a reservoir from which eventual progressive resistance will emerge. And so while we don't know a whole lot about the cell biological and molecular nature of these cells, there are a few features of residual disease cells that do seem to be observed recurrently in different studies by different groups involving different cancer types even. So for example, of course it makes sense to say that Residual tumor cells have evaded cell death. So they're not undergoing cell death in the face of a drug that's supposed to kill them. That's a defining feature of residual disease cells. They very frequently exhibit cell cycle arrest or slowed proliferation. They very frequently, independent of the tissue that they arise from, often exhibit a shift toward more mesenchymal differentiation. We see certain reproducible epigenetic, transcriptional, and metabolic changes in residual cancer cells. So think, for example, about an upregulation of oxidative metabolism. We see hypermutability in certain models of residual disease. We see multi-drug resistance, so these cells are not just resistant to the drug that's been selected, that they've been selected against, but with others as well, oftentimes. And there's, in the last few years, it's emerged that a real hallmark feature of these cells is an increased vulnerability to ferroptotic cell death. So while these are relatively vague, they are nevertheless a starting point for thinking about what might be unique dependencies in residual disease. And that's kind of what we did. What we said is, well, could we anchor our thinking around one of these characteristic features of residual disease and try to build out an understanding of dependencies of residual tumors based on that anchor point? And so we really chose mesenchymal differentiation as our starting point. Because again, this is a highly recurrent feature of residual tumors. So the question that we asked is, are residual tumors vulnerable to mesenchymal selective dependencies? In other words, if cancer cells that have entered a mesenchymal state develop unique dependencies because of their entry into that state, then perhaps those vulnerabilities might be at play in residual disease. And so there was this extraordinary student in my lab named Shane Killarney who took on this project, and he came up with a really creative idea. He went into DepMap, and he took all the epithelial cancer cell lines in DepMap, which is like 800 and something, and he gave each cell line a transcriptional score depicting where that cell line is on the continuum from the most epithelial state to the most mesenchymal state. And then what he did is he sorted those lines from most mesenchymal to most epithelial, and then he asked which dependencies extracted from CRISPR genome sequencing screening data are most aligned with the mesenchymal state relative to the epithelial state. So are there selective dependencies? And when he does that analysis, he identified, reassuringly, the one gene we know to be a mesenchymal selective dependency, which is GPX4. This was discovered in a couple of Nature papers a few years ago. And he also found a cassette of additional genes that score more strongly than GPX4. And one of them is this relatively understudied so-called dark kinase known as PKN2. And so we got interested in PKN2 for a number of reasons. One is that PKN2, the signaling functions of PKN2 are not well understood. Second, it's a kinase, so it's druggable. Third, in the developing mouse embryo, if you knock out PKN2, you actually ablate the development of the mesoderm, which provides in vivo evidence that there might actually be a mesenchymal selective vulnerability at play. And finally, if you conditionally delete PKN2 in an adult mouse, the mice are viable, and suggesting that there may be a therapeutic window for future PKN2 inhibitors if they exist. And so to validate this finding, we first took a panel of epithelial-like cancer cell lines and mesenchymal-like cancer cell lines, and we found that knocking out PKN2 has no effect in the epithelial lines, but a big effect in the mesenchymal lines. This is not just a correlation. The relationship with mesenchymal state is causal. We know that because we can take different models, isogenic models of epithelial to mesenchymal transition, and we can see that the PKN2 dependence emerges after cells undergo an EMT. So for example, these are melanoma cells that undergo something like an EMT as they develop resistance to a BRAF inhibitor. So the parental cells are here, the BRAF inhibitor resistant cells are here. And what we see in these cells is that when they go from parental state to resistant state, or in other words, into a more mesenchymal state, they develop PKN2 dependence. We see this in multiple models, for example, normal epithelial cells cultured with TGF-beta and other things as well. So this is just one example. So this is a real mesenchymal selective vulnerability. To understand what PKN2 might be doing, we used gene coessentiality analysis. So many of you may know that if you look at genes that travel together in genetic screens that are performed either across many cell lines or across many perturbation conditions, what you tend to find is that genes that travel together are often involved in the same biological pathways or processes. And so we said, what are the genes that travel with PKN2 through DepMap? And if you look, what you see is that there's this set of genes that travel very closely with PKN2 that are all involved in hippo-YAPTAZ signaling. And so what that suggested is that PKN2 might regulate hippo-YAPTAZ signaling. So to ask that question, we first knocked out PKN2 in 293T cells, and what we saw was increased activation of the hippo pathway as seen by phospholates and phospho-YAP, and loss of total TAZ levels because the hippo pathway suppresses YAPTAZ. So you activate the hippo pathway when you knock out PKN2, and you suppress YAPTAZ. We see the same thing at the transcriptional level. So this is a PKN2 target gene, and if you knock out PKN2, and sorry you can't see it right here, but what you get is suppression of this target gene. You can also go in the opposite direction. So you can express in 293T cells a version of PKN2 in which the N-terminal autoinhibitory domain has been cleaved off the protein. And if you do that, what you see is that the hippo pathway is suppressed and YAP and TAZ are activated as visualized by loss of phosphorylation of YAP and stabilization of TAZ. And with that, you also see, and I'm going to point over here now, a upregulation of a canonical YAP-TAZ target gene. Okay, so PKN2 is necessary and sufficient for modulating YAP-TAZ signaling. And finally, if you take a mesenchymal-like cancer cell line and you knock out PKN2, the fitness defect from that insult is fully rescued by a mutant form of TAZ that is hippo-independent, that is constitutively active independent of the hippo pathway, and it's not rescued by a version of TAZ that is under control of hippo signaling. And so what this all suggested is that PKN2 is regulating the hippo pathway, and that in turn is regulating YAP and TAZ. To understand how this was working at a more granular level, we did phosphoproteomic analysis. So we took cells, we introduced an activated version of PKN2, and we looked at new phosphoepitopes in the proteome. We found a bunch of new phosphosites, and then we overlapped those new phosphosites that we found with proteins that contain a motif, or phosphosites that contain a motif that is thought to be a direct PKN2 substrate motif. And the way that we got that substrate motif was through data from a recent paper from Ben Turk and Mike Yaffe and Lou Cantley, which systematically tried to define phosphomotifs in kinases across the kinome. And if you do that, you get these, you can subselect for these red dots, which are putative direct PKN2 target sites. And if you look at if you look at those and you then further down select for proteins that might be involved in the YAP-TAZ pathway, or the hippo-YAP-TAZ pathway, you see one protein, this protein SAV1 at residue serine 90 that is phosphorylated in the presence of active PKN2 and that is a known canonical component of the hippo pathway. So it turns out that indeed PKN2 can directly phosphorylate SAV1 at position S90. That phosphorylation in an in vitro kinase assay we lose if we incubate with a version of SAV1 where there's a point mutation at position S90 that can't be phosphorylated. PKN2 can also phosphorylate SAV1 at position S90 in cells. We see that with a gel shift assay where you can see phosphorylation of SAV1 in the presence of active PKN2 but not inactive PKN2, and that phosphorylation goes away if you introduce an S90A mutant. We're not really real experts with AlphaFold 3, but we know some folks who are. we worked with them to look at the interaction or potential interactions between PKN2 and SAV1. And what we were really struck by was that there are multiple contact interfaces between these proteins. They bind actually quite snugly. And although it's difficult for you to see here, the serine residue at position 90 sits right in the kinase domain of PKN2, making it seem really plausible that this could be a direct substrate. And perhaps most importantly, we were able to show that if you introduce a version of SAV1 that can't be phosphorylated at position S90 into cells, and then you simultaneously introduce a hyperactivated PKN2, you block the ability of PKN2 to stabilize TAZ. So here is in the presence of wild-type SAV1. This is in the presence of S90A mutant SAV1. And furthermore, you block the induction of YAP-TAZ target gene expression by activated PKN2. So here you activate YAP-TAZ target gene expression when you introduce active PKN2, and here we show that you reverse that effect when you introduce a point mutation in SAV1 S90. So where does all this get us? Well, it suggests a model whereby in an epithelial state, PKN2 is inactive, the hippo pathway is thus active, YAP-TAZ are shut down. But when a cell undergoes a mesenchymal differentiation event, PKN2 becomes active, it phosphorylates SAV1 at position S90. This leads to inhibition of hippo signaling, activation of YAP-TAZ, and YAP-TAZ-driven target gene expression. Now, if you recall, the reason why we started studying PKN2 was not because we wanted to define a new component of the hippo pathway, it's because we wanted to look for gene dependencies that are selective to mesenchymal cancer cells. Because we believed, based on the literature, that residual cancers tend to exhibit upregulated mesenchymal differentiation, and thus mesenchymal selective vulnerabilities might also be residual disease vulnerabilities. So now we turned our attention back to that question. The first thing we did is we took a bunch of different oncogene-driven cancer cell models. We cultured them for 18 days in the presence of their cognate-targeted therapies, and we measured the expression of both mesenchymal state markers and YAP-TAZ target gene probes, or target genes. What we found was that in these 4 models of EGFR mutant non-small cell lung cancer treated with an EGFR inhibitor, osimertinib, and these 2 models of KRAS G12C-driven non-small cell lung cancer treated with a KRAS G12C inhibitor, and in these 2 models of BRAF mutant cancer, this melanoma, this colon cancer, treated with BRAF plus MEK inhibition, that almost in every case, we see upregulation of both mesenchymal gene expression and YAP-TAZ target gene expression. So consistent with the hypothesis that when you go into residual disease state, you upregulate mesenchymal gene expression and you upregulate YAP-TAZ signaling. We also see biochemically that if you look at phosphorylation of the activation loop of PKN2, which is thought to be a surrogate for kinase activity, in some of these models, what you see is that the time-dependent activation of PKN2 kinase activity that really perfectly correlates with the activation of TAZ signaling. To understand if the real key question, which is PKN2 important for the survival of residual disease? What we did is we cultured EGFR mutant lung cancer cells with EGFR inhibitor to send them into a residual disease state, and then we either knocked out a control or knocked out PKN2, and then we looked for the ability of the cells that remained to grow out following withdrawal of targeted therapy, basically a measure of whether residual disease cells survived. And when you do that experiment, you see a really, I think, compelling result. So cells in which no gene has been knocked out, you see a degree of survival and then regrowth following the removal of targeted therapy. But in the setting of induced PKN2 knockout, you see that cells are unable to grow out following the removal of targeted therapy, suggesting that we've eradicated many of the residual disease cells. To understand a little bit more about what's going on here, we did this longitudinal experiment where we introduced osimertinib, the EGFR inhibitor, we induced PKN2 knockout, and then in residual disease cells or DTP cells, we measured signaling, apoptosis, apoptotic signaling, apoptosis at the cellular level, YAP-TAZ signaling, and cell cycle. And then we also left osimertinib on the cells for an extended period of time and measured the ability of cells to re-engage the cell cycle in the residual disease state. And what we found was really consistent with our expectations. So PKN2 is phosphorylated when cells reach the DTP state. The knockout of PKN2 leads to an upregulation of caspase cleavage, an upregulation of annexin 5 positive cells, suggesting that some cells are dying because of PKN2 loss, suppression of induced YAP-TAZ signaling. And while there is initially no effect on the cell cycle in the early phase of DTP, because cells aren't cycling at all, what PKN2 knockout does do is it blocks the ability of cells to re-enter or begin slowly re-entering the cell cycle when you leave them, the drug on them for an extended period of time. Now that's great. That's a bunch of data in one model, which is the same model that I'm showing here. It's PC9 EGFR mutant non-small cell lung cancer cells. So to expand this result, we did an experiment that looks like this. We knocked out PKN2 or a control, and we looked at the ability of cells to survive targeted therapy treatments after an extended period of time. PKN2 clearly blocks that ability without having much of an effect on the parental cell lines on their own. But you can really extrapolate this finding to many other models, and that was really exciting for us. So in here, models of BRAF mutant melanoma and BRAF mutant colorectal cancer, PKN2 blocks residual cell survival. Here in KRAS mutant models of non-small cell lung cancer, same thing here and here. And here in the bottom four, in models of EGFR mutant lung cancer, same thing. You see diminished survival in the setting of PKN2 knockout in residual disease. Now, interestingly, after we submitted this paper for publication, a friend of mine at UCSD wrote me an email. And he said, my lab a year or so ago was studying a drug called darovasertib. Darovasertib is a marketed PKC inhibitor that's used for the treatment of uveal melanoma, and it looks like is going to be approved by the FDA soon following the results of a pivotal ongoing phase 3 trial. It turns out, though, that darovasertib, this marketed PKC inhibitor, is also a potent PKN2 inhibitor. It wasn't designed to be, but it is. So you should test whether this drug works in your residual disease models. And at least in early studies, it looks like it does. So here, if you look at survival following osimertinib therapy, we see a degree of survival that is lost in the presence of darovasertib in two different models, here and here. And I'll just tell you, while I'm not showing the darovasertib only control experiment here, darovasertib on its own has no effect on these cells. It's like water. And interestingly, also has a really good safety profile in patients, consistent with the genetic knockout studies in conditional adult mice. In vivo, you can create EGFR mutant lung cancers. You can allow them to grow alone or in the presence of PKN2 knockout, and they grow just fine. If you treat with an EGFR inhibitor, you send them to a residual disease state. The depth of response is deeper when you knock out PKN2, as you'd expect. And importantly, if you then remove the targeted therapy, the cells can regrow in the absence of targeted therapy as long as PKN2 is present. But if PKN2 has been knocked out in the tumor cells, they lose their ability to regrow. So that's an example of some of the work that we're now doing in the setting of residual disease. I'm now going to shift gears a little bit to a different phase of disease, which is the setting of acquired resistance. And I just want to do a time check here. So what time does the seminar end? Oh, perfect. Okay. The clock on this thing is different. Okay, perfect. So I do have time. So in the setting of resistance, one of the things that we know, that I'm sure all of you guys know, is that over the last few years, people have started doing studies where they look at the presence of potential resistance mechanisms in patients who have begun to progress on treatment with targeted therapies. And what people have seen is this really overwhelming and daunting thing, which is that in these patients, there's usually not evidence of just one resistance mechanism. There is usually evidence of many resistance mechanisms that are all occurring at the same time. And that's a big challenge for the field that, frankly, I don't think the field knows how to solve. Because if you tried to use a cocktail of drugs that block all the resistance mechanisms. First of all, we don't have such drugs, but even if we did, it's very, very likely that you run into major toxicity problems. But if you just block one or two resistance mechanisms, it's also very, very likely that the disease would evolve around those drugs because other clones aren't driven by those resistance mechanisms. And so we've been trying to think, are there creative approaches that we might be able to use to take on this really complicated problem of resistance? So our thinking in this area really got stimulated by a study that we published in the lab about 7 years ago by Lauren Crawford and Katie Singleton. And what this study showed, it was a study that took place in the setting of models of BRAF mutant melanoma. And what it showed is that while there are many different resistance mechanisms that can and do drive resistance to BRAF inhibitors or BRAF plus MEK inhibitor therapy in BRAF mutant melanoma, and while these different mechanisms function through different effector signaling pathways, that there is a key event that seems to always happen, and that is the hyperactivation of MYC. And this activation of MYC in multiple, really a wide diversity of models, is both necessary and sufficient for the development of resistance. Okay? So what this study suggested was that at least in the setting of BRAF mutant melanoma, there is convergence of many different resistance mechanisms onto a single downstream effector that's really critical for resistance. One of the ways that we really developed confidence in this idea is that it's known that when MYC becomes hyperactivated in cells, a bunch of other things happen. It rewires the metabolism, it rewires the transcriptome, and so new, predictable, MYC-dependent synthetic lethalities should emerge. And in fact, that's what we observed. So we observed that in models of resistance driven by many different mechanisms, we always saw MYC-dependent synthetic lethalities arising. A few years later, two students in the lab, Kevin Lin and Justine Rutter, made a similar observation in AML cells. So what they were studying in this paper were AMLs treated with a BRD4 inhibitor, which is a known dependence that they were trying to exploit in AML. When they evolve cells to resistance against a BRD4 inhibitor, this is a panel of different cell lines, each one where we're looking at the parental cells and the resistant cells. What we always see is hyperactivation of MYC in the resistant cells relative to the parental cells. So it's very analogous to what we saw in the melanoma case. That is notable because these different models are driven by distinct resistance mechanisms. So it's not like there's the same resistance mechanism that's leading to MYC activation. It's different resistance mechanisms that are all leading to convergent MYC activation. And again, consistent with what we showed in the prior study, when MYC is hyperactivated in these resistant tumor cells, they develop new dependencies. One of the most famous synthetic lethal dependencies that's known to occur in cells that hyperactivate MYC is they become more apoptotically primed. And so in this study, what we showed was that when cells develop resistance to a BRD4 inhibitor, they have hyperactivated MYC, and because of that, they also have substantially increased sensitivity to a BCL2 inhibitor called ABT199 or venetoclax. And this increased sensitivity that they observed is fully MYC dependent. And I'm not showing the data here, but you can go in and modulate MYC, and you can block this induced dependency. And we see this not just in cell culture models, but we also see this in vivo models. So here, for example, we took an AML PDX. We took cells from that model and we measured their sensitivity to ABT199, which is a BCL2 inhibitor, again, in both the naive state and in the state in which they've relapsed on BRD4 inhibitor treatment. And what you see is this substantial sensitization of resistant cells to this drug. So if you kind of put these findings together, what it suggests is maybe a little bit of a new model of how to think about and maybe target drug-resistant tumor cells. So instead of thinking of tumor cells as initially being present in an oncogene-driven state, and then once resistance emerged, looking like an explosion of heterogeneity, maybe instead it's useful to think of tumor cells as existing in one of two distinct states. So state 1 is a state driven by the original driver oncogene. And state 2 is a state where you are resistant to inhibition of the driver oncogene, but as a result, you have a new sensitivity that's based on some convergent phenotypic changes that the cells are undergoing. In the case of the first two studies I showed, these changes would be driven by MYC. So this idea might or might not sound plausible to you, but I'll tell you one other way that I make sense of it. You know, when resistance emerges, even though there may be many different resistance mechanisms at play, all of the cells that we care about share an important phenotypic property, and that is they have developed the ability to proliferate in the presence of a drug that should otherwise kill them. And so essentially what we're reasoning here is that if they have that one shared phenotypic property, the ability to grow in the presence of a drug that should kill them, maybe they also share a constellation of other similarities, of other shared properties, which could include shared collateral sensitivities. So we wanted to explore whether this idea might be more generally extrapolatable. And so what we did is we took models in the lab of acquired resistance to different therapies. So for example, we took AML cells with acquired resistance to a BCL2 inhibitor. We took melanoma cells with acquired resistance to a BRAF inhibitor, colon cancer, head and neck cancer, and lung cancer cells with acquired resistance to EGFR inhibitors, and so on and so forth. And then for each of these drug-resistant models, we tested its sensitivity against a panel of about 30 drugs that we hand-selected to target diverse aspects of cell physiology. And what we were really looking for were drugs that were more potent against the resistant cells than they were against the parental cells from which they arose. So when we see that, we denote it with a blued circle here. So this is the sensitivity of the resistant cells normalized to the sensitivity of the parent cells. When we see a red dot, it's the opposite. It is cross-resistance. In other words, the resistant cells are resistant to both the drug they were selected against and to whatever the new drug is that's represented by the dot. So what this analysis showed us is that the idea of collateral sensitivities, or fitness trade-offs if you'd prefer, in drug-resistant cells, this is not like a rare thing. Every single time, virtually, that we do this experiment with only 30 drugs, we find examples of collateral sensitivities. So on the basis of this, a number of years ago, Kris, a student in the lab, said, let me do a deep dive. Let me see if I can actually find a druggable collateral sensitivity that might have some real robustness. And then if I can understand how it works. I will preface this part of my talk by saying this is really still an open area of investigation lab. This is not a complete story, but it's an interesting story, I think, that's pointing in some interesting new directions. So Kris chose for his project to work on EGFR mutant non-small cell lung cancer, and he did that for really a simple reason. We knew a lot about the mechanisms that drive resistance to EGFR inhibitor therapies. And we had good models, both in our lab but also in the broader community, of mechanisms of resistance to EGFR inhibitor therapy. So what Kris did is he assembled a panel of 3 drug-resistant models. One that had a T790M mutation that renders it resistant to first-generation EGFR inhibitor therapies, another that has IGF-1R hyperactivation, and another that has MAPK1 amplification. So all 3 of these models are resistant to EGFR inhibitors, at least first-generation inhibitors. And what he did is he tested the sensitivity of these models to a library of drug compounds, and he compared that sensitivity to the sensitivity of the parental cells from which these models arose. And what he looked for are drugs that are always more potent against drug-resistant models than they are against their parents. So a shared collateral sensitivity. When he did that experiment, these are the results, and I'll actually point over here. He showed that while there are lots of examples of cross-resistance, many of these are tyrosine kinase inhibitors like EGFR inhibitors, there were a relatively small number of strong collateral sensitivities. And of 2,100 compounds in this library, there was exactly one compound that showed collateral sensitivity against all three of our drug-resistant models. And that was this compound known as MCB613. And so Kris became pretty interested in this molecule. And the next thing he did is he wanted to know if this shared collateral sensitivity actually is robust. Do we see it in model after model? We see it in these three models, but do we see it elsewhere? And so the first thing he did is he took an EGFR mutant lung cancer cell line that is sensitive to an EGFR inhibitor, that's this curve here, and he evolved resistance, and he picked clones, and he extracted a bunch of drug-resistant clones against a first-generation EGFR inhibitor. And what he found was that, as you'd expect, these clones are all very, very drug-resistant. Now, if you take those same cell lines and you test their sensitivity to MCB613, what you find is that in every single case, they are also more sensitive to MCB613 than their parents are. So even though presumably these represent a diversity of resistance mechanisms, they all show sensitivity to MCB613. We did the same thing in a second cell line, evolved resistant cells, and then measured their sensitivity to MCB613. Again, they all seem to be hypersensitive. And then finally, we took a patient-derived EGFR mutant lung cancer cell line, evolved resistance to osimertinib, the third-generation EGFR inhibitor, and then showed that those cells again exhibit collateral sensitivity to MCB613. So at that point, we said, this seems like a pretty robust phenotype, and it's worth deeper investigation. Before I get to that mechanistic insight, I'll show you some interesting games that a drug like this allows us to play. So, the idea behind collateral sensitivity is really this. If there are drugs that are always potent against drug-resistant cells because of a fitness trade-off that drug is exploiting, then we ought to, in principle, be able to combine that drug with the original drug that you used to treat that cancer and make a drug cocktail that has a really unique property, which is that it selects against resistance. Because the initial drug is selecting for drug resistance, and the second drug is specifically potent against drug-resistant cells. And so it should make it very hard for cells to evolve. This is an experiment that sort of reflects that. So if you take parental EGFR mutant lung cancer cells and you mix them with several different drug-resistant daughter cell lines, and then you treat with either MCB alone or gefitinib alone, you see the relatively quick outgrowth of cells. In the case of gefitinib, that outgrowth reflects the drug-resistant cells emerging. In the case of MCB613, that outgrowth reflects the parental cells outgrowing. But if you combine these two drugs together, now all of a sudden it becomes very, very difficult for resistance to emerge. In fact, so difficult that you can remove both drugs at some point and no cells grow back. Now, MCB is not a drug, although I've referred to it as such. It's really a probe compound. It has really poor pharmacological properties. But if you work really hard, you can formulate it to get it in vivo as much as possible, which is still not great. We've done the PK studies. But you can do it enough to show that while parental EGFR mutant lung cancer xenografts are relatively non-responsive to this compound, those are the black curves, a drug-resistant model, which are the red curves, are quite responsive to the compound. So at that point, we wanted to understand what this compound might actually be doing at the mechanistic level. So we took a look at what the compound looked like. And if anyone in here is a chemoproteomics expert or a medicinal chemist, you are probably suppressing the urge to vomit right now, because this is really a horrific compound from the perspective of medicinal chemistry. And I think one of the reasons why this molecule is problematic, or at least potentially problematic, is that its structure looks like a potentially promiscuous electrophile. So this molecule can, in principle, participate in two distinct Michael addition reactions, one taking place here, one taking place here, where nucleophilic amino acids can, in principle, react with this molecule. And so when we first looked at this molecule, I said, Kris, this is really concerning to me, because it could be that this molecule is killing resistant cells because of some general electrophilic stress. And it also could be that this molecule is working through many different targets and not just one. And neither of those are things that I'm super excited for us to chase down. Consistent with the idea that this might be a problematic molecule is the fact that different studies that have described research with molecules that look like MCB613 often identify different putative mechanisms of action, right? Consistent with some degree of pleiotropic activity. But Kris really wanted to soldier forward. And so what he did is designed an approach that was really rooted in proteomics, functional genomics, and biochemistry and chemical biology to try to really tease apart what this molecule might be doing in cells. So the first thing he did is he recognized that he could install at this position on MCB613 an alkyne handle, and that this new derivative compound exhibits roughly the same biological activity as the parent MCB613 compound. It is still selective for drug-resistant cells. It has about the same potency. And he also realized that he could identify a very similar compound to MCB613 that has no biological activity. So it looks very similar, but it doesn't have activity at all. And he could similarly install an alkyne handle on this compound. And if he does this, he can incubate these two different compounds with cells, and he can use click chemistry to conjugate these compounds to beads and pull down the proteins that interact specifically with MCB613. So if you do that, you get a list of a few hundred proteins. And then what you can do is you can create a custom CRISPR library of guide RNAs that target the proteins that interact with MCB613. And this is really based on the idea that if MCB613 is inhibiting a target, then knocking out that target ought to phenocopy the drug itself. We didn't know, by the way, that what the drug was doing was inhibiting the target, and I think we now know that what it's doing is probably not exactly that, but nevertheless, it was a reasonable starting point. So we made this custom CRISPR library, we did screens across the drug-resistant cells and the parental cells, and we found one really interesting gene that was the top scoring gene in the screens, and that's KEAP1. So KEAP1 in our screens was always more potent when knocked out against resistant cells than parental cells. It hurt resistant cells more. Sure enough, you can validate that MCB613 really does, and this is the daughter compound of MCB613, really does interact with KEAP1, so you can pull down KEAP1 with these click chemistry enabled compounds. If you knock out KEAP1 in cells, and then you culture them over time, you see that the black parental cells survive just fine, but the red drug-resistant derivatives have a more pronounced fitness defect. Interestingly, this fitness defect is not as strong as the defect we see with the drug, and I'll be talking about why we think that is. And then lastly, if you knock out KEAP1 in drug-resistant cells, and then you take the cells that have survived, and then you treat them with MCB613, you find that they have no sensitivity to MCB613 anymore. So what all of this means is that MCB613 binds to KEAP1, and that KEAP1 is required for the toxicity of this drug. So this was an important step forward. To further understand what might be going on, we said, well, maybe MCB613 is a KEAP1 inhibitor. And so what we did is we assembled a panel of other molecules that have been described in the literature as KEAP1 inhibitors. One such molecule is curcumin, which is interesting to us because not only is a putative KEAP1 inhibitor, but structurally it resembles MCB613, namely in the sense that it too has two potential reactive electrophilic groups on it. We also looked at a panel of other drugs, some of which are covalent electrophiles, all of which have been described as KEAP1 inhibitors, but which do not contain that symmetric, dually reactive motif. And when we tested these drugs for their potency against our resistant and parental cell lines, we observed something interesting. MCB613 and curcumin both behave the same way. What I mean by that is, the more resistant a cell line is to gefitinib, the more sensitive it is to either MCB613 or curcumin. But if you look at all other putative KEAP1 inhibitor compounds, this relationship breaks down. So only curcumin and MCB613 do this. To understand a little more what might be going on, we looked at the effects of MCB613 on KEAP1, and what we observed is that it causes destabilization of KEAP1. So in a dose-dependent manner, if you add MCB to KEAP1 in purified cell-based assays, you see a thermal shift denoting destabilization. If you do the same experiment in a thermal shift assay in cells, you see the same thing, destabilization of the molecule by MCB613. So this was also distinct from other KEAP1 inhibitors because all the other KEAP1 inhibitors that we tried, with the exception of curcumin, are thought to be KEAP1 stabilizers, not KEAP1 destabilizers. So at that point we thought, well, maybe what we have on our hands is we may have happened upon a KEAP1 degrader, and that this molecule is working through degradation of KEAP1. And when we looked at a Western blot, we saw evidence that was consistent with that. So as you add more and more MCB613, you see less and less KEAP1. But then we took a look at the entire membrane that we were running this Western on, and we saw that while indeed you do see a loss of the monomeric form of KEAP1, you also see correspondingly the emergence of what appears to be a dimeric KEAP1 species. It's exactly twice the molecular weight of monomeric KEAP1. And I'll tell you, just to relieve the suspense, that this species really is dimeric KEAP1. We've done the mass spec. Interestingly, and consistent with the idea that this might be a covalent reactive compound, we see that the dimeric species that forms is really, really stable. We can't denature this thing, suggesting that it's a covalent interaction. Now, does this matter? Does it matter that you're losing the monomeric species and you're gaining a dimeric species? Well, actually, let me stop for just a second. I got ahead of myself. The presence of a covalent dimer, coupled with the known structure of MCB613, suggests, as I mentioned before, that what might be happening is that MCB613 is acting as a covalent molecular bridge, connecting two KEAP1 monomers together. And all of a sudden, our finding that curcumin phenocopies MCB613, whereas monoreactive electrophiles don't, makes a little bit more potential sense because curcumin can also participate in principle in two distinct Michael addition reactions. So to really understand if this dimerization was important, what we did is we made a daughter compound that's similar to MCB613 in every way, except it can only participate in a single Michael addition reaction. It cannot covalently react with two different KEAP1 species. So we said, look, if the dimerization matters, then this compound will tell us. As you might expect, when you add MCB613 to cells, you shift KEAP1 from a monomeric molecular weight to dimeric. When you add this daughter compound, ARM143, which can't participate in two different reactions with the molecule, you only get a monomeric species. I will mention that this species is covalently liganded by ARM143. We've tested that. But most importantly, what we find, and I'll show it over here since there's a little glitch here, is that ARM143, unlike MCB613, has no activity in cells, okay? So now what we've done is we've shown that a molecule that looks like MCB613, but is only different in the sense that it can only modify KEAP1 one time and can't form a covalent dimer, loses its ability to exhibit this cool collateral sensitivity phenotype. To understand better how this might be working, we focused in on the domain structures of KEAP1. And what Kris literally did is he chopped up KEAP1 in lots of different ways to try to isolate the domains that might matter. Ultimately, what these studies revealed, and I'm not showing them for brevity, is that the BTB domain is really the domain that matters the most. And that's not surprising. KEAP1 has a native dimerization domain, which is the BTB domain. And so it sort of makes sense that covalent dimerization might also require that domain. He looked in the BTB domain, he scanned it for potential nucleophilic amino acids, and then he substituted each one of those amino acids with an alanine that can no longer participate in a Michael addition reaction And what he found was a single residue, K97A, that when introduced into KEAP1 blocks the overwhelming majority of oligomerization of KEAP1. I just want to point out one wrinkle in this experiment. In order to do this experiment, we ectopically express KEAP1. And when we ectopically express KEAP1 in cells, we don't just get a nice dimer the way that we see when we have endogenous KEAP1 in cells. We also see a bit of a laddering effect, suggesting that there are also some higher order species that are being created. We don't really know yet what these species are, but when I say that K97 ablates the effect, what I really mean is it ablates the formation of all these species to about, 90%. And importantly, if you introduce full-length KEAP1 into cells where you mutate only this one residue, lysine 97, you can completely block the sensitivity of cells to this drug. Okay? And importantly, and I'll touch on this point later on, the K97A mutant of KEAP1 is not turning on NRF2. So the backstory here is that we were worried that maybe KEAP1's ability to rescue the toxicity of the drug might be related to the fact that this mutant is turning on NRF2, and that's causing some kind of a general redox pro-survival program. But we don't see evidence of that in cells. Now, if you dock a molecule of MCB613 into a dimer of KEAP1, where one of the dimers is in blue and the other one's in green, what we see is something interesting, which is that from a geometric perspective, this molecule fits really beautifully into a cavity that's formed between two lysine 97 residues. So geometrically speaking, there's nothing that says that this couldn't actually ligand two distinct KEAP1 species and create a covalent dimer via lysine 97, though to be super clear, we haven't proven that that's what's happening. Now, in thinking about KEAP1, we had to think about NRF2, and I mentioned NRF2 a moment ago. But there had been a paper that was published about a year ago that described in lung cancer cells a phenomenon known as reductive stress, where they argued that if you activate NRF2 in certain lung cancers, you could kill them through reductive stress. And so one of the concerns we wanted to eliminate was the possibility that NRF2 activation was the mechanism of action of this drug. Now, we didn't think that was the case because other KEAP1 inhibitors don't have the same activity. But we did test it formally and showed that while MCB613, as you would expect, turns on NRF2, NRF2 is dispensable for its activity. So to show that, what we did is we knocked out NRF2 in cells and treated them with MCB. And what we found is that if anything, you modestly sensitize cells to the compound, but you don't confer resistance to the compound. So the reason why this compound is killing cells is not because of activation of NRF2. Okay, I only have a few more minutes, and what I'm going to try to finish up with is a little bit of understanding of what's happening downstream of KEAP1 dimerization that's causing this selective cell death effect. And this is a place where in the project we only have fragmented understanding. So one of the things that was known about MCB613 is that it can cause activation of an integrated stress response in cancer cells. That was described in the first paper that was published that studied MCB613. And in fact, in our early studies with this molecule, we saw the same thing, which was activation of the ISR. We didn't really know what to do with that information until a really beautiful paper came out from Doug Green's lab in Cell. And what this paper showed in the exact same EGFR mutant lung cancer cell lines that we had been studying is that when you treat EGFR mutant lung cancers with an EGFR inhibitor and you send them into a residual disease phase, they upregulate ATF4, a component of the ISR. And that upregulation of ATF4 is required for the survival of these residual cells. And so we said, there might be something to this. And so the first thing we did is we just looked at ATF4 levels. And what we find is that, consistent with the Green group's work, while parental EGFR mutant cells have relatively low levels of ATF4, our drug-resistant cells have high levels. And this is just one drug-resistant cell line, but we see this across many different resistant models. Now, what is interesting about ATF4 is that it can act in two different ways. If there's a little bit of stress that a cell encounters, ATF4 can be activated to help to repair that stress. But if too much stress is encountered, ATF4 can become super activated and in turn activate a CHOP-dependent cell death process. And so since we knew that MCB613 turns on the integrated stress response, including ATF4, we reasoned that perhaps the high basal levels of ATF4 in these cells might become super activated by MCB613. And indeed, that's what we see. So if you look at a much lower exposure gel in our drug-resistant cell lines, we see that MCB can potently activate ATF4 as well as CHOP, and that the activation of ATF4 and CHOP are both required for cell death. So if you knock out ATF4, you block cell death by this drug. If you knock out CHOP, you block cell death induced by this drug. So just to kind of pull that together, I realize that that's a complicated story. I think what our results are pointing to is something like this. In EGFR mutant non-small cell lung cancer, if you add an EGFR inhibitor, you get residual disease cells. These cells are able to survive because they have hyperactivated ATF4. That's the work of Doug Green's lab. If you continue to add an EGFR inhibitor, our results suggest that you expand a population of resistant cells that have vestigial, leftover, high ATF4 expression. As a result, these cells are very sensitive to MCB613, a compound that somehow, through an undetermined mechanism, following the dimerization of KEAP1 leads to ATF4 superactivation and ATF4 and CHOP-dependent cell death. There are lots of questions that are still going on in this story. We're really interested in understanding how ATF4 oligomerization is really driving the ISR. We don't know the answer to that. And we're really interested in trying to figure out whether MCB613 might be something that we could optimize for clinical studies. But just as I wrap up, I think what I would like to kind of conclude with is this idea that we, like many others, are interested in tumor dependencies. But what we think is really interesting is the idea that number one, in the setting of residual disease, we think that many of the hallmark features of residual disease, most notably the mesenchymal differentiation that's present in residual disease, might impose mechanistic vulnerabilities on these cells. And that while there might be some clonal heterogeneity in these cells, the fact that these cells broadly share a phenotype, in this case, a growth-arrested mesenchymal-like state, might mean that they will share a vulnerability to a drug that exploits that phenotype. And similarly, in the setting of resistance, while there are heterogeneous resistance mechanisms that are at play, the fact that many different resistant cells all exhibit a common phenotype, which is the ability to proliferate in the presence of a drug that should be killing them, they also harbor shared vulnerabilities that can be exploited to make drug therapies that select against resistance through collateral sensitivity. So I think I showed, while I was talking, the people who were doing the work that I was really highlighting. These are other folks in my lab who are all doing their own interesting things that I didn't talk about today. And then, of course, our funding sources. And with that, thank you for your attention. I'm happy to answer any questions. Speak loud, or I can repeat your question.
Thanks for the great talk and for all the compelling evidence.
Thank you.
So for your first part of the story, Fralin, do you know if PSM2 directly phosphorylated? And also, everything was found in cells, right?
Yeah.
In the actual tissue,
Yeah.
there's a lot of contact inhibition.
Yes.
People, do you think that can translate to the baseline of people activation in the tissue?
Yeah. So there were two questions. The first question was, do you know that PKN2 phosphorylates SAV1 directly in an in vitro kinase assay? The answer is yes, and we actually show that in the paper. It does. In fact, that's the only place that phosphorylates SAB1. The second question was, in a real tissue where you have contact inhibition, you have cells in a closer geometric confinement, do you still see hyperactivation of the Hippo pathway in the residual disease setting? And the answer is we do. And we see that not only in animal models, but other folks have seen that in humans with residual disease. It's interesting. One of the things that's beginning to emerge is the idea that in tumors that have been substantially depleted by a therapy, their extracellular mechanical environment is different. They find themselves in a much more stiff environment than they were when they were in a kind of a soft, squishy tumor, and that might actually also further contribute to Hippo activation, or sorry, to YAP-TAZ activation in that setting. So the answer is we do see it. Now, do we see it in every residual cell? Is there Hippo activation and PKN2 dependence? I don't think we know the answer to that question. And that's an important question that we want to try to suss out. Yeah. Yeah, it's a good question. So there is a drug that we think is a PKN2 inhibitor called darovasertib, which I just mentioned very briefly in the talk. If it turns out the darovasertib really is an in vivo PKN2 inhibitor, it'll be a great probe for these types of questions. We're still not totally sure. When we do experiments with darovasertib, we see a lot of phenocopying of PKN2 knockout, but we don't always see perfect phenocopying. There are cases where we don't see the same effects, and we don't really know what's up with that. Pardon me. And I noticed that when the company that makes darovasertib, when we talked to them about our findings, they were a little bit unsure as to whether they actually believed it was a PKN2 inhibitor, despite biochemical evidence that it appears to be. So I think the jury's still a little bit out as to whether that's the case. Yeah. 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 That's really interesting. No, we never did. No one's ever mentioned that, actually. We'll look at that. Yeah, but how does, so NRF1 is ubiquitinated by KEAP1. So when KEAP1 is inhibited, you would expect it to go up, right? And then that in turn, you would expect to activate ATF4? Yeah, that's really interesting. Wow, interesting. Okay, interesting. Thanks for mentioning that. That's really interesting. We'll see. Yeah. Oh, sorry. Yes. Yeah. Great question. So our initial analysis was with all epithelial and melanoma cell lines, because all of those undergo mesenchymal differentiation in the setting of residual disease. We see PKN2 coming out from a pan tissue analysis, but we also then took the one tissue that's most represented in DepMap, which is lung cancer. We did the same analysis just within that histology, and we see the same thing. So I think it seems to be pretty universal, and consistent with that, we have validated mesenchymal specific PKN2 dependence across models from probably 5 or 6 different lineages. Yeah. And you had a question? Yeah.
Very compelling story.
Thank you.
I was just wondering, do you still see a converging point in all your stories? Because it seems like each of the story is making one hero, but do you see any kind of converging point in all the 3 stories? Yeah.
So the question is, do we see convergence in the biology that all these different stories are showing us? Right now, I think I could hand wave it, but I'm not sure that we have compelling evidence of that. But this is the direction my mind works in. I tend to think like if there are shared phenotypic properties of the cells that are most problematic, then there have to be shared dependencies. Or maybe they don't have to be, but it seems like there would likely be. And so I think within a defined context, so let's say resistance to an EGFR inhibitor through whatever mechanism you want, it does seem like there are commonalities. Whether those commonalities further extend to both resistance and residual disease, or to resistance and residual disease in other tissue contexts is unclear. There was a while in my lab where the joke was that Kris thinks MYC is the driver of all resistance. And I kind of did. I don't necessarily anymore. So I don't think we have evidence of that right now.
For example, if you have Nick over the cell has intensity that is more high because of the high translation rate. So I was wondering, like, find generate theme on that.
Yeah. Not yet. Not yet. Yeah.
Hi, thank you for your excellent talk.
Thank you.
And the gene you mentioned in the residual disease part seems works very well. And what do you think is future application of that gene?
Of PKN2?
Yeah.
Yeah, so I think PKN2 is really interesting as a potential drug target. So the only other way that we can inhibit YAP-TAZ, I mean, as you know, what we showed is PKN2 is working in residual disease because it's turning on YAP-TAZ. So then you might reason that, well, why don't you just use a drug that inhibits YAP-TAZ? Well, there's a problem there. There are a new class of drugs called the TEAD inhibitors, which TEAD is a cofactor that's required for YAP-TAZ to drive gene expression. And so if you use a TEAD inhibitor, you can block YAP-TAZ signaling. And in fact, those drugs do have amazing activity in residual disease models consistent with our findings. The problem is TEAD inhibitors inhibit YAP-TAZ independent functions of the TEADs, and they don't inhibit YAP-TAZ functions that are TEAD independent. And so what you see with TEAD inhibitors is substantial toxicities, particularly renal toxicity in patients. With darovasertib, we don't see that renal toxicity. Similarly, in the conditional mouse knockout model of PKN2, there were no renal toxicities that were reported. So I think that PKN2 could be a really interesting target for any cancers driven by YAP-TAZ activation, or at least for many cancers driven by YAP-TAZ activation, including residual tumors, not just because it works, but also because it might be better than the only other strategy we have to block YAP-TAZ, which is the TEAD inhibitors. So I think that's the angle that we're thinking a lot about, therapeutically, Yeah.
Thank you very much for a very good talk.
Thanks.
I have a question about the hypo-independent function of YAP and TAZ that has been reported, and we know that they contribute to the resistant mechanism. If you ever explored more like a subcellular localization changes that happens independent of hypo?
It's a great question. We are exploring that right now. To be more, I guess, complete here, we're exploring really two possibilities. The first possibility is that, at least in some mesenchymal cancers, that PKN2 may be regulating YAP-TAZ in addition to the SAV1 mechanism, also through Hippo and SAV1 independent mechanisms. We don't have compelling evidence that that's happening, but we think it's important to understand because PKN2 is associated with Rho GTPases and cytoskeletal remodeling, and it just makes sense that it could. The second thing we're exploring is the possibility that PKN2 might also have YAP-TAZ independent functions. We don't know what they would be, but we were exploring the possibility that might be the case for kind of the same reason, cytoskeletal remodeling and things like that. But we don't have any data yet on that. So right now, everything we know seems to be explainable through SAV1, but I don't think that we're yet convinced that that's always going to be true. Yeah. Okay, thanks.
Okay, everybody, if I could just make an announcement before you go out. If you agreed to join the luncheon with Dr. Wood, it is happening in conference room A across the street. So you can make your way over there now. Thank you.